Welcome to Philsa Hub Research University, your ultimate guide on your next research project.
We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the Philsa Hub team of research scientists and some invited subject matter experts. Philsa Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment.
And the best part? It's absolutely free.
Visit us at philsihub.com and be part of this learning journey.
Let's learn and succeed together here at Philsa Hub Research University. Welcome to Philsa Hub Research University, your ultimate guide on your next research project.
We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the Philsa Hub team of research scientists and some invited subject matter experts. Philsa Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment.
And the best part? It's absolutely free.
Visit us at philsihub.com and be part of this learning journey.
Let's learn and succeed together here at Philsa Hub Research University. Welcome to Philsa Hub Research University, your ultimate guide on your next research project.
We will lay down the best practices in research ideation, literature review, experimental design, and data analysis to guide you as you carry out your research project. Learn from the Philsa Hub team of research scientists and some invited subject matter experts. Philsa Hub Research University will be your personal coach through every stage of your research project, from ideation to final assessment.
And the best part? It's absolutely free.
Visit us at philsihub.com and be part of this learning journey.
Let's learn and succeed together here at Philsa Hub Research University.
Magandang umaga po sa ating lahat. Welcome to the Filipino Science Hub. Thank you for your continued support to this online platform. To our new participants, thank you for checking us out today, and we would like to officially welcome you to the Philsa Hub family. So bago po tayo magsimula, I would like to share some house rules in order to facilitate a smooth session for everyone. For our Zoom participants, we would appreciate it if you could turn your audio and video cameras off during the webinar. If you have questions or comments, feel free to type them on our Zoom chat box or YouTube live comments, and we will try to accommodate as much questions and comments as possible. After the talk, we will have a 30-minute Q&A where the Zoom participants will have the opportunity to ask their questions live. We would also like to request for everyone to not record the webinar. The full video of the session will be posted on our official YouTube channel. For those who would like to receive a certificate of participation, a Google form link will be posted on our Zoom chat box, YouTube live comments, and YouTube video description towards the end of the webinar. Please make sure that you enter the correct information, especially your email address, to ensure receipt of the certificate. Also, for you to obtain the certificate, you will need to subscribe to our YouTube channel as well as follow our page on Facebook. Please note that we receive hundreds of requests and it takes around 7 days or so to process all the certificates. If you do not receive your certificate after a week, please feel free to send us a message on Facebook or email us at philsihub@gmail.com. So again, welcome po sa Philsci Hub. Welcome to all our Zoom participants and our YouTube live viewers. For those who are new and may not be familiar with who we are and what we do, allow me to share our mission and vision in the next few minutes. Filipino Science Hub is an online platform founded by our CEO, Filipino scientist Dr. Jeffrey Camacho Bunkin, back in 2012. Currently, we are a registered nonprofit organization based in Houston, Texas, United States. Our mission is to promote STEM education and the culture of research among students and teachers in the Philippines and abroad. Our current global leadership team is comprised of volunteer Filipino scientists and seasoned STEM educators from different parts of the world. Our vision is to see a technology and innovation-driven Philippines, and we believe that this can be achieved by creating a new generation of well-rounded Filipino STEM enthusiasts. This vision is being carried out via our two major programs, the Philsci Hub Ed program and Philsci Hub Research University. Through Philsci Hub Ed, we are able to empower and support our teachers when it comes to strengthening STEM fundamentals education. We believe that this is important because the more equipped our STEM educators are, the more they can inspire our students to pursue a career in science, technology, engineering, and mathematics. Through this program, we have been able to serve our teachers by creating STEM teaching modules as well as conducting teachers training webinars, all of which are accessible on our website for free. So we have also been able to collaborate with major educational organizations that have the same goals and advocacies as Filipino Science Hub. Through these partnerships, we have been able to further expand our reach and help more students and teachers, especially during the time of pandemic. So together, everybody achieves more. The other major program that drives our vision forward is Philsci Hub Research University. We believe that in order to strengthen a STEM culture, students also need to be able to learn how to conduct a scientific research. As we know, many scientific and technological breakthroughs happen through research, and we would like to equip our younger generation by providing them proper scientific research training. So ika nga po, start them young. Here at Philsci Hub Research University, we provide free courses on fundamentals of scientific research, such as research ideation, literature review, how to write a research proposal, how to communicate your research work to the scientific community, among other courses. Through this program, we are also able to bring practicing scientists closer to the STEM educational sector. So because of this closer contact, our students become more aware of the possible STEM career paths available for them and how these careers contribute to the advancement of the society around them. So up to date, we have been able to reach over 70,000 students and teachers all over the world, and we know that this will continue to grow in the years to come. And this is why we are all very excited on what is in store here at Philsci Hub. Our official website is www.philscihub.com. This is where you can access all our materials such as webinars, tutorials, modules, virtual lab, and special features. We are also present in several social media platforms such as Facebook, LinkedIn, YouTube, and TikTok. So follow us wherever you're most active on to get updated with our web events. So again, thank you for being with us this morning. And to officially start our webinar, I would like to turn the virtual floor over to our host for today.
Okay, magandang umaga po sa inyong lahat. I am Jeffrey Bunkin and I will be your host for today. All right. Okay, so let me share my screen first. If you guys can just give me a minute. Kamusta po kayong lahat ngayong araw ng Sabado? Medyo nakamute nga pala sila. All right, so
naman po, sir.
all right, yeah, maraming salamat po sa inyong pagsagot. Ayan, so inaayos ko pa lang po yung screen that I will be sharing with you. So if you can bear with me. Maraming salamat po sa muli ninyong pagsama ngayong araw ng Sabado, lalong-lalo na sa ating mga estudyante galing sa Bataan National High School. So you guys turned out, nandito na naman kayo para samahan kami. So we hope that you actually learn a lot from today's session. So so this is the second installment of Filipino Science Hub's Biology Month. So hanapin ko lang yung aking. Siguro po we'll ask for people po to for the participants to mute their mics if that's possible. All right. Laging nagma-magic yung Zoom yung Zoom ko, maling screen yung na-share. All right, so can you guys see my screen? Mama, can you see my screen? All right, so magandang umaga po ulit sa inyong lahat. So I will start with a reintroduction of Filipino Science Hub. This will be very quick. So Filipino Science Hub, as mentioned in the introductory video by Dr. Dindy Voiles, who happens to be the treasurer of Filipino Science Hub. So Filipino Science Hub is a non-profit organization registered here in the United States, and it is currently being led by Filipino scientists, innovators, educators, as well as business professionals. And the goal of the organization is to ultimately help increase the number of Filipino students who would pursue STEM careers in the future. So that's the reason why sobrang masaya kami sa session ngayon, kasi majority of our participants are high school students. So hopefully this session will further inspire you to pursue STEM, or to develop yourselves in your interest areas in STEM. And then at the same time, hopefully, like as we introduce Filipino scientists to you, magkaroon kayo ng idea ano ba yung klase ng trabaho na pwede ninyong ma-experience if you would specialize in STEM. And today we are actually going to be talking about aquatic mycology. So this is a very exciting field. So all right. So that's Filipino Science Hub. So what do we intend to do? So ang primary objective of Filipino Science Hub is develop education technology and learning materials and training programs para sa lahat ng mga estudyante. And when we say lahat ng mga estudyante, the goal of the organization is to come up with strategic interventions para kumbaga, start them young, no? So preparatory level pa lang magkaroon na tayo ng intervention para ma-kondisyon ang mga utak nyo na STEM is actually the place to be. Yan. So all the way up to the college level. So we are coming up with programs para ma-instill yung interest sa STEM. And then hopefully, at one point, kapag dumami na yung portfolio ng materials namin, we can also come up with supplemental materials that you can help in the mastery of STEM subjects. So yan. But hindi lang kami dyan tumitigil. What's very crucial for our success is to also foster the development of your STEM teachers in parallel. So, dinedevelop namin yung students, but for this effort, or for all of our programs to become sustainable, dapat along with it, we should also be helping build the capacity, or we should be able to empower yung ating mga guro. So kaya mapapansin nyo sa audience ngayon, it's a good mix of students and teachers. So yan, so sama-sama tayong mag-grow along this process. And at one point last week, I actually shared with you the main reason why, or the real life challenge. So the current challenge is that the Philippines is lacking scientists and innovators. So UNESCO made this recommendation in 2018 that for the Philippines to sustain its growth and for it to be able to keep up with its neighboring Southeast Asian countries, it has to have at least 380 scientists or innovators per million Filipinos. Unfortunately, in that year of 2018, we only had 189, wala pa sa kalahati. Napag-iwanan talaga tayo ng mga kapitbahay natin. Singapore, ang density ng scientists per million of its population was 6,900. An order of magnitude higher than what we have. Malaysia had 2,100 per million Malaysian. Thailand had 975 per million Thais. So ang layo ng gap. So kailangan nating mahabol yan, and we have to start sa basic education. Kasi dyan sa inyo, sa mga estudyante sa elementary at sa high school, sa inyo magagaling yung future scientists at mga leaders sa field. So I'm hoping, so magmumukha akong saramplaha kasi I will never get tired sharing this information. All right? So please keep this in mind, most especially when sa mga nasa junior high, when you decide on where to go for your college specialization, please consider STEM. All right, so ano ba yung mga ginagawa namin? So sabi namin meron kaming mga interventions para matulungan kayo in your mastery of STEM courses. So magbibigay lang ako ng ilang mga examples. So over the past couple of years, we have been coming up with laymanized and Tagalog English lectures in different STEM areas. And these are tutorial videos brought to you by Filipino scientists. So people who are experts in the field and people who live science on a daily basis, we are the ones teaching this. So check all of these out kasi these could be like the most basic, and I think quite proudly so. Ito yung pinaka-relatable siguro na pagkakaturo ng mga STEM topics para sa inyo. So these are just examples of some of our tutorial courses sa chemistry topics. So I actually develop all these. I work hard to put together all of these materials. So check all of these out. And then over the past couple of years, we have also developed supplemental STEM modules. Ito, pwede itong gamitin ng teachers kasi meron itong teaching guide part. And then meron itong mga problem sets and challenge questions. So ito mga supplemental STEM modules na ginawa namin na to, meron ito sa chemistry, merong topics in biology and physics as well as earth science. So yan, you can navigate, you can actually access all of these materials sa aming social media spaces. It would be most convenient to navigate our website or our YouTube channel as well. So we recommend that you check all of these out. So these are materials created and curated by math and science teaching majors from the University of the Philippines, Los Baños. So sobrang pinili din namin yung mga taong gumawa ng mga materials na to. And then over the past couple of years as well, we have been producing home-based experiments and essentially 1 to 2 minute science videos then to help inspire yung students, no? Ano ba yung magagawa yung actualization ng natututunan nyo sa klase kapag kayo nasa bahay. So we have all of these materials. Meron kaming naka-upload sa YouTube, and I think I don't know if you guys are aware, we are also on TikTok. So we had this partnership with TikTok Philippines, which we call Learn on TikTok. So we had developed 1 minute STEM videos. And I think like in totality, yung 15 videos that we had rolled out last year, they were viewed a total of about 3 million times. So you should check that out. Kung gusto niyo ng medyo magkaroon kayo ng sense of inspiration or at saka kumbaga bite-sized materials para matutunan niyo yung STEM. So this is actually a good way of learning them. Hindi nagpakita yung attachment ko on TikTok. Anyways, so these are supplemental materials. But then now, as Professor Dindi mentioned earlier, we also have a portfolio called Philsa Research University. So for most of you in high school, you have your capstone project. So that would mean that you will have to perform a research project. So Filipino Science Hub, under the leadership of Mr. JP Onya, who's our vice president, we had put together a portfolio of research of training materials on each step involved in the research process. So from ideation, all the way down to thesis writing, scientific article writing, and conference presentation. So lahat yan, if you are unfamiliar of the basics, you guys have to check that. You guys have to check this out. And ito, hindi to mga boring na lectures kasi mapapanood niyo, you can experience the training na para lang kayo nakikinig ng chismisan ng mga scientists. So ano to? Oo, medyo marites at maritos ang mga ang nag-deliver nito sa inyo. And Dr. Connie Gibas is actually one of the people responsible for some of these content. So we'll talk to her later, in a bit. And then and then ito, yung session ngayon is part of what we call a research university special topic. So what we do is that we invite Filipino scientists trained in different STEM areas and from different based in different countries in the world. And what we do is we invite them to talk about their focus areas, like their areas of specialization. So for today, you're gonna have an idea of the career journey of our speaker, what he does in his chosen field of specialization, and hopefully towards the end, students take advantage of your opportunity to ask him questions if you really want to, to deepen your understanding of today's subject. All right, so last na for reminders about like Filipino Science Hub. So we're present in almost all social media spaces. We have an official website called with URL www.philsahub.com. We're also on YouTube. We just hit 14,000 subscribers today. So, for you to be able to get notifications, just follow us on our official channel. We're also on Facebook. That's where we have like our most of our following. And then we're also present on TikTok. So we're also making our presence known in areas na nandun ang mga kabataan. So nagtitiktok na rin kami. All right. So for today, again, we would like to welcome you to Filipino Science Hub's Biology Month. And today we will have the second installment of a series of 4 talks. And I would like to call in my co-host for today, Dr. Connie Givas, who is actually the brain behind this. So Dr. Connie Givas was the very first biologist to be featured on Philsa Hub and to become a member of the leadership team of Filipino Science Hub. And palagi kami nag-uusap. I call her Mama Connie because we met while I was in grad school and pinapakain niya ako dun sa bahay niya. So please pardon me, baka kayo magulat kayo, tinatawag ko siyang Mama. So like Dr. Connie Gibas really wants to instill sa mga kabataan kung ano yung kahalagahan ng biology as a field of study in biology. And it's a very big area, right, Mama?
Yes.
Yeah. And so gusto niyang ibahagi sa inyo ang kanyang love for biology, and we really had to think, siya nag-isip talaga siya ng panel ng speakers that we would feature today that would give you an idea of fundamentals of biology all the way up to its applications. Ayan. So Mama, could you also make a comment siguro, no? Bakit natin ginagawa itong Biology Month?
Kasi this is important kasi para makita ng mga estudyante, marinig nila yung all aspects of biology. So hindi lang basic biology, kundi yung application, hindi lang microbiology, meron ding organismal, meron ding cell biology. So we have the cancer drug discovery. Tapos meron ding organismal, yung mag-uusap tayo tungkol sa mga carabao in the last webinar. So this is all aspects of biology na para makita nyo, hindi lang ang biology, it's not only about microbiology. There's cell biology, there's organismal biology, and there's molecular biology. So lahat yan. So hopefully, dito sa series of webinars na to, meron naman kayong mapupulot kahit papaano.
Ayan. And so, for today, ang topic natin is about, our speaker will introduce you to the area of aquatic mycology. And to do the honors of introducing our speaker for today, Dr. Connie Givas will introduce our speaker.
Okay, hello again mga hubbers. So ito na naman tayo sa pangalawa nating session. So ang speaker natin ngayon ay si Mr. Mark Calabon. Si Mark, he did his Bachelor's of Science degree sa West Visayas State University sa Iloilo. And then he did biology, magna cum laude. And then he went on to get his masterals at the University of the Philippines, Visayas in Miagao, Iloilo. And then after that, he went to the Mae Fah Luang University in Thailand, in Chiang Rai, Thailand, to work on his doctoral. And malapit na siyang matapos, mga 2 buwan na lang. So we would like to welcome Mr. Mark Calabon. Come on, Mark.
Yan,
Hello,
Sir Mark, take it away.
Okay. Good morning, everyone. So I will share my screen. Okay. Okay na po ba, sir?
Yes, sir. But wala pa po sa presentation mode.
Oh,
Yeah,
okay
there
na
you
po ba,
go.
sir?
Yes,
Okay.
yes.
Good morning, everyone. Anong oras na sa Thailand? So 9:30 na pala. Okay, my topic for today is about aquatic mycology. But aquatic mycology, it's a really, really broad concept, or I mean a subject or a topic. So what I did is parang nag-overview na lang ako ng methods that I will be discussed today and the recent taxonomic classification of aquatic fungi. So as you can see on the screen, these are aquatic fungi in their natural habitat. So I will discuss this further in the next few slides. Fungi are ubiquitous in nature. When we say ubiquitous, we can find them anywhere, everywhere. Minsan, pag hindi natin hinugasan yung rice cooker natin, makikita natin molds or yung mga filamentous fungi nandun sa pagkain, and some, they can cause spoilage also ng foods or fruits. Or minsan, pag
Mama ko si TJ.
Okay. And for example, if you're trekking, for example, in a mountain or on your backyard, maybe you can find plant pathogenic fungi. I will show you the laser pointer. This is a plant pathogenic fungi. Maybe you can also find Ganoderma lucidum in your backyard, or dung fungi, or when you do gardening, for example, it's very good in our mental health nowadays during pandemic, and you can see mycorrhizal fungi in your loam or humus, or it can cause diseases in animals or maybe also in us, in humans. But the question is, nakakita na ba kayo ng fungi in aquatic environments? Maybe wala pa. So today, that's my specialization. I study on aquatic mycology, but mainly focus on the taxonomy and phylogeny of aquatic fungi. So kami yung nagdi-discover ng new species, new taxa, or nagdi-discover ng fungi with a comprehensive description, and using molecular data, we find the phylogenetic placement of that certain taxa. So aquatic fungi can be found anywhere. For example, sa marine environment, sa mangrove, or either in the coastal areas, coastal dunes, or in salt marsh environment, or even from freshwater environment. This is Bohol, and this is in Antique. This is a stream, and in the lake also. The diversity of aquatic fungi is quite amazing, actually. And we are looking forward that we can provide a comprehensive and detailed morphology and phylogenetic analysis. And I will discuss some of them here in my talk today. So yeah, that's my topic. I will discuss the overview of methods in studying aquatic fungi, and I will discuss to you my Ph.D, which is the taxonomic classification of aquatic fungi. my journey, bago tayo pumunta kung saan ako nandoon, I need to reminisce my journey para naman to honor my mentors, especially in the Philippines. I started my degree in West Visayas State University in Iloilo City under the advisory of Professor Crisanto Lopez Jr. We study antifungal activity of mushroom, Ganoderma lucidum extract on the fungal population of stored peanuts. So parang yung curiosity ko that time parang nag-spark. Maganda pala yung fungi pag nasa colony, pag nasa media, and you can see the sporulating structure. I did not know that time that it can cause diseases sometimes, but yeah, it's so beautiful. So when you plate the fungi, parang, amazing, the color, the structure, and how from the mature stages to the immature stages to the mature stages, parang different yung morphology and exciting. I did not proceed to medicine, and I decided to do my master's in UP Visayas under the advisory of Professor Resurrection Bisadaba. He is an expert in mangrove and any fungi. Yeah. And they're also from the project of Dr. Wilfredo Campos. So we study sponge-associated fungi, and we are lucky enough to publish this on an ISI journal that time. And here, since Dr. Sadaba, Dr. Sadaba, professor, is asking for a Ph.D. student from the Philippines. Actually, two of us here. One is working on mushroom taxonomy and cultivation, and I am working on the taxonomy and phylogeny of aquatic fungi. My advisors are Professor Kevin David Hyde. He is a world-renowned aquatic mycologist, but also in other field of mycology. His mentor is also now my mentor also, Professor Evan Benjamin Gareth Jones. He is one of the pillars of aquatic mycology under the supervisory of Ajahn Saranyapat Gunmi from Thailand. So that's my journey. I was inspired to work out. When I saw the colony of fungi, I was inspired, and I said, yeah, maybe this is my future. And yeah, see where I am now, studying the taxonomy and phylogeny of aquatic fungi. The outline of my talk will focus on the aquatic fungi from its definition, from the history, the classification, and I will discuss how do we study aquatic fungi. When I learned that most of our participants are high school students, so I need to teach them how to study aquatic fungi. So maybe under like a special problem in the high school senior program, they will be inspired and they can continue to study aquatic fungi in the Philippines. After that, I will do the overview of the classification of freshwater and marine fungi. The data that I will share to all of you are actually nearly published and nearly to be published we are proofreading the data. And I will discuss also the recent progress and gaps per taxonomy and per area of aquatic mycology. So what is an aquatic fungus? By definition, there are different category and the presence in water. When we say resident, they can be indwellers or immigrants. When we say indwellers, the metabolic activity, or they can stay in the aquatic environment for maybe in their whole life. They can be obligate fungi also in aquatic environments. Some of them are immigrants. When you say immigrants, these have an extra aquatic nature. It means that they can live in terrestrial environment or in the nearby plants. For example, in the mangrove area, some of them are not submerged, so some of them are in the upper part of the mangrove, and they can be considered as migrants or versatiles. And the presence in water, indwellers are permanent, so they are obligate. Migrants are periodic; they have periodic activity, and the versatiles are irregular, so they are not permanent. The activity is they are constant. For the indwellers, periodic and sporadic. It means that the metabolic activity, they can do their functions properly in the aquatic environment, or in periodic, they can be, periodic fungi, they can be periodic or sporadic. For irregular, this is the irregular. They can be sporadic or they don't have any activity at all. For the transient, these are actually fell down. For example, there's a trunk fell down on the water. Maybe they don't have activity after all and they cannot sporulate, or they will just die. So that's why they are transient fungi. The definition of freshwater fungus, we will classify them based on the salinity. We have the freshwater environment, the marine environment. Freshwater fungus, according to Thomas, is any fungus that can live whole or part of their life cycle, that can rely on the different substrates and aquatic environment. But marine fungus, traditionally, they are classified into obligate or facultative marine fungi. When we say obligate, they can live only in the marine environments and they cannot function outside that one. For the facultative, they can both be like immigrants fungi. They can live from freshwater or marine environments or terrestrial environments. So in 2016, the marine mycologists convened with to find out what's really the definition of marine fungi, because marine fungi, sometimes when you say marine fungi, parang kina-consider nila na, oh, dapat obligate lahat or facultative. But how about those fungi, like for example, that can be found anywhere, ubiquitous, for example, Penicillium and Aspergillus, they are very, have a worldwide distribution, they can found everywhere. Can we consider them as marine fungi? So they convene and they come up with a general and very good description of what marine fungus is. So marine fungus, or any fungus that is recovered repeatedly from marine habitats, so they can be considered also as marine-derived fungi, for example. One of the criteria is that they're able to grow and sporulate in marine environments. They form symbiotic relationship with other marine organisms, or they can adapt and evolve at a genetic level or be metabolically active in marine environments. So we will use this kind of description when we study aquatic fungi. And for example, I focus on the classification of aquatic fungi when we review the publications and how we consider that, for example, the species can be marine. So we need to be careful with the materials and methods, and for example, the phylogenetic placement of certain taxons. So let's go to the history of freshwater fungi. First, it was in 1880 when the first Hyphomycete was discovered. No? This is Heliscus lugdunensis by Saccardo. And after that one, in 1893, de Wilderman discovered 4 new species of freshwater Hyphomycetes. 3 of these have a tetraradiate conidia, and 1 has a filiform conidia. And here, you can see here, these are the species name. And in 1942, parang hindi kinonsider ng mga marine mycologists that time yung discovery ni Wilderman. So after 50 years, when Ingold discovered the habitats that the marine aquatic fungi or freshwater fungi were discovered, and they can sporulate there. And he observed CT Ingold, Cecil T. Ingold. So yeah, he discovered 16 species. 10 of these are new. The discovery of Ingold in the natural habitat of aquatic fungi was considered to be the minor mycological industry. When you say minor mycological industry, it's paved the way to more studies on the occurrence and geographical distribution of aquatic fungi. And there are early works after that one, and I will not discuss more, but these 2 are very important. For marine fungi, the first species was discovered in 1849. This is Typicula typharum from Taifa, and Speria posidonia from Posidonia seagrass, oceanica. Early works, there are a lot of mycologists, but I cannot discuss all of you because they have ample amount of contributions in the field of marine mycology. So what I can discuss to you now is in 1944, there's a discovery of fungal community in driftwood from the intertidal habitats, and Buckhorn and Linder were responsible for that one. And one of the most important thing happened in marine mycologists in 1966, where they convened to have a marine mycology symposium, and a lot of marine mycologists that time shared their knowledge on marine mycology, and collaboration after that one. And currently, I'm here under the supervisory of Evan Benjamin Garrett Jones, Professor Hyde, and we are working on marine fungi. So these are important persons in the field of marine mycology. We have Professor Sparrow, Professor Johnson, Professor Jones, this is Professor Jones, Colin Mayer, and etc., etc. This is Professor Hyde. These are some of the pictures in Hong Kong long time ago when they started the study of Asian mycology here in Asian mycology. So for the methodologies, I just wanted to have an overview for the high school student. So we collect the samples in the natural habitat. For example, we collect the different substrates. We do it, incubate it. We observe it using the macro and micromorphology of the fungi. And we get the characters of the fungi. That is very important in the description, especially when you want to introduce a new species, new genus, new family, or new order. We did single spore isolation also. So we will make sure that what we are isolating is aquatic fungus, not from terrestrial environment. We incubate the culture to grow, but aquatic fungi are very slow growing. So we need to be more careful because they are easily to be contaminated if we are not careful enough. So we did photoblade preparation for the description. This is very important, especially our center in Thailand. We are more of morphological and phylogenetic analysis, and we are very little strict when it comes to the morphology and the photographic plane. We did the colonial description, and for the colonial description, we also get the morphology and culture. So this is very important when you want to have a sexual and asexual morph connection of aquatic fungi. And after that, we do molecular analysis to find the phylogenetic placement. And this is also very important also if you want to introduce novel taxa or new species to science. So these are the substrates that we usually observe. So you can see here, I'm so sorry. Here we have in the marine environment, we collect wood. For example, there are also from the mangrove habitats, seeds, pneumatophores, also from the prop roots, leaves, submerged, etc., etc. It should be submerged because if it's not submerged for a quite long time, we cannot isolate aquatic fungi. And for us, because we already know the morphology of aquatic fungi, we can distinguish, oh, this one is an aquatic fungus because of this belongs to genera. We also isolate from salt marsh environment in Thailand and also from UK. Sediments also. You can see here, this is a fruiting body of fungi. And also from freshwater environment, we collect woods and grasses. I'm so sorry. Parang hindi next. Okay. After that one, when we collect the samples, we do morphology. So very important here, we use compound and dissecting microscope to obtain the micromorphology and the macromorphology of the fungi in their natural habitat. We don't do plating. Usually when I did my master's, we do plating, but here we need to observe them in the substrate because they have different morphology and usually they have the different morph stages. And after that one, I can draw. So for example, we observe, this is a fruiting body. We checked for the fruiting body, we cut it. So when we cut it, we can determine if it's a sexual morph. Just take note, this is sexual morph or the teleomorph. This is what we call, this is Acilomycin, sorry, cannot see. No? For the asexual morph or the anamorph, we have the hyphomycete. And this is the Silomycy. So what's the difference? Acilomycete, these are the following structure. They have the asci, ascospore, and the whole ascomata. For the hyphomycete, we have the conidiophore and the conidia. For the Silomycete, we have conidiogenous cell and the conidia and the conidiomata, the whole fruiting body. I will not further discuss this one because it's a long discussion, but I just wanted you to see the different morphology of aquatic fungi. And after that one, we cut with the razor blade manually. So vertically, or the cross section of the fruiting body. And for hyphomycete, we just pick it up. For the ascomycete, the asexual morph, we cut it and put it in the slides. So we put water, put the fruiting body there. Okay, so you should be careful we don't have bubble, because if you have a bubble, it's difficult to see the morphology and you cannot see the whole character of the aquatic fungi. Okay, well, sorry, it's not going to the next part. What is this? Okay, after that one, when we do the cutting of the observation, we observe in the microscope, and these are the morphology. So it's quite interesting, right? So here is the hyphomycete. You can see the conidiophore. It's like a small forest on a very small wood twig. And you can see the morphology of the conidia. This is the conidia. See the spore. Long time ago, we just see the spore as like globus or circular, things like that. But in the natural habitat, they have different morphology. This is a Silomycete. Silomycete, you can have the whole fruiting body here. This is the conidiomata, the wall, and you can see the conidiogenous cell. This is very important when you see when you wanted to have a detailed morphological description. And this one, we synonymize, we introduce this as a new combination. This one we introduce as a new species. This is Halobysothidium from the genus Halobysothidium. This one from Sodobactrodesmium. So here, for the dissecting microscope, you see the morphology in the wood. For the compound microscope, you see the micromorphology of the aquatic fungi. For the this one is ascomycete. This is the sexual morph. This is very important also when you want to observe the different stages. For example, the immature to mature stages of the asci. These are the spores of the fungi. Here, different species also. These are new species that we introduced from Halobysothidium and from Sarcophora. So here again, the morphology in the dissecting microscope is here. And for the compound microscope, you can see the very beautiful morphology of the ascospore and other morphological characteristics. When you observe already, you need to obtain culture. One way to do that is to do single spore isolation. From the word itself, you need to only obtain one spore that is germinated and transfer this to another new Petri dish. It's very important when you want to have a good molecular data. So what you're gonna do, just mark the Petri dish, cut the section, the fruiting body, pick it up with a sterilized needle, pick these are the spores, put some water, this is sterilized water in the sterilized what is this? I forgot the name, with a cover slip. Glass slide. Sorry. And this one, you need to put all the fruiting body here, the one that you cut. Crush it to release the spore. So pipette it out. You need to pipette it out and put in your gridded Petri dish or media plates. So incubate in room temperature for 1 day, but for aquatic fungi, usually it takes 2 to 3 days for them to germinate because they're very slow growing. And sleep. I will sleep that night. After the next day, we need to observe. And for example, we have germinated spores. You can see here, you need to observe in the compound microscope. If you found it, go to, transfer it to another fresh medium. If you have no spore germination, just cry, choke, and you don't have data, just cry. But the thing is, you need to use another different culture media and repeat the process again. That's quite tricky and very depressing also when you work on aquatic fungi. But it's okay. It's an interesting field. Sorry, these are already germinated spores. So you can see here the different germ tube. Why are we taking pictures of germinated spores? This is very important when you want to see you want to see the pattern of where the germ tube arises. For example, this one, it arises from the basal part. Some of them will arise on the different angle, and some at the polar and at the apical and basal cell. So this one, you need to pick it up, cut again. These are the spores, and you need to be more careful because sometimes some aquatic fungi have a lot of fruiting body also in the substrate. So you need to familiarize yourself of what you are observing. So we need a mutiform spore, right? This is not the cylindrical one, so you need to pick this one up. And you need to be careful also because sometimes maybe if you're not good enough or maybe you're tired and you will pick this up because they're very small. You can see only using microscope. So you need to be more careful. And after that one, take the picture and transfer to another plate. When you transfer to another plate, this will be the picture. Quite interesting. So for me, because they're slow growing, I put 4 or 6 in one colony. And you can see here, this one are aquatic fungi with the same morphology. And this one also, this is another like a pure culture already of aquatic fungus. You can see also the sporulating structure here. After that one, we do the photographic plate description. So we need to be concise with the morphology we wanted to share. For example, if you want to publish, this one is a new species. So if you want to publish it in a good journal, you need to have a good morphological description also. And one thing we need to do also is to observe the culture. And this is very important when you want to have a asexual morph connection of aquatic fungi. So these are the cultural observation. When we say cultural observation, we observe them from the culture. These are the chlamydospore. This is a new species, Cladophila aquatica, we introduced. And this is a new host record, Sauriella nipae. You can see in the culture here, these are the cultural micro characters. It's quite the same, but you can see the different stages from mature to immaturity. See, they become hyaline, then they become like dark brown at the end, pigmented already. And after that one, when you have a good enough mycelia already or culture, you can do a phylogenetic analysis. This is just a step, but this is very easy also. So you have DNA sequences, you need to blast to see the closest relative. You need to assemble the forward and reverse data. You need to assemble your multi-gene data set. You edit it, you do alignment, you cut it using Chumal, you do conversion for further analysis. This is for the maximum likelihood, Bayesian, and the maximum taxonomy. And after, if you have this one, you can have a phylogenetic tree already. And you will visualize, revise. You do not revise it like revise the whole tree, but you only revise the name. Sometimes when you do analysis, you have the code, right? So the correct name of the fungi. And you do further analysis to determine if it's new or not. So taxonomic and phylogenetic analysis of aquatic fungi contribute to what we have current understanding of what the classification of aquatic fungi. It's the advent, the incorporation of molecular data in the morphological characters of aquatic fungi has actually resulted in a modern classification system for kingdom fungi. And now, last 2011, mycologists convened for a one fungus, one name concept. It means that there is no like dual nomenclature already, but and only have one species name for a particular fungi. This is very true. For example, Penicillium and Aspergillus. Penicillium is the anamorphic or the asexual stage, and the Penicillium is the sexual or the teleomorphic stage. And mycologists wanted, like, it's very difficult when you run phylogenetic analysis and there's a Penicillium, another clade is Penicillium. So why not name them as one? Because they are a sexual and a sexual morph of each other. So they choose Penicillium, the same stage also with Aspergillus. Neosartoria is the sexual morph. Aspergillus is the asexual morph, and they choose priority, the rule of priority. They choose the Aspergillus because Aspergillus is very important, in nature, ecologically and medically. So they choose Aspergillus. But usually, if, for example, there's a cases also that mycologists choose the sexual morph form rather than the sexual morph form. But it's case-to-case basis, and there's a lot of criteria for that one. And there are a lot of published work and rule for the naming of fungi and for choosing what name, the classification you should use. So these are the new species that I introduced. I will not discuss them one by one, but I just wanted to share with you that you can do it also if we have a good enough funding and a good enough supervision. Okay, so this is interesting. I just wanted to share. This one, which we will publish soon under proofreading. This is Pseudophyllocephala Amygdala. This species was considered in Leiotomycetes long time ago. This is Phylocephala clade because Pseudophyllocephala was formerly known as Phylocephala ubicula. But when we did run the analysis, we observed that the placement of Phylocephala ubicula was not on the Phylocephala clade in Leiotomycetes. Leiotomycetes is a class of Ascomycota. When we ran a big tree, we found out that Phylocephala ubicula clustered with Chaetospiralis and different class, Sordariomycetes. So we introduced a new genus, Pseudophyllocephala, and synonymize all the taxa that clade with the Pseudophyllocephala. So yeah, so here are the synonymized or transferred taxa. And I found out that there is a species from our group that was formerly known as Phylocephala ubicula, but I told him that maybe your species from previously known as P. ubicula is a new. So we observed the specimen again and obtained the molecular data. Because it has a very different conidial morphology, we introduced this as a new species and supported by molecular data. This is very basal to other species of Pseudophyllocephala. See, this is globus to subglobus. And this one is like cuboidal, rhomboidal, rhizoid sometimes. And see, it's like a triangle. Very different. And another one, this genus doesn't have molecular data, and I found in a freshwater habitat. This is Microinterlobium aquadetusporium. We place it in Pleosporales, but it's in Certae because it's very different when it comes to morphological details to Testudinaceae. So we introduced this one in inserting studies Pleiosporales, but we will obtain more data and we can introduce perhaps new family because it's very different from Tesudinaceae. So let's see for that one. And for the phylogenetic analysis, there are different studies of freshwater fungi and marine fungi. When it comes to Sordariomycete, this is a class of Ascomycota in freshwater habitat. My senior, Weidong, have a monograph, not a monograph, but all the morphological and whole phylogenetic analysis of that class. Dothidiomycete, Sordariomycete Zonglong is also my senior. They have this whole class phylogenetic analysis of Sordariomycetes and Dothidiomycetes. Currently, we updated the families of Sordariomycetes. You can access the publication, Refined Families of Sordariomycetes. This is very important when you want to know the different placements and the updates from the previous families of Sordariomycetes last 2013. And Dr. Hong Sanan also published refined families of the Dothidiomycetes that will be a reference also when it comes to the taxonomic classification of this class. For the marine fungi, we have different revision also. So you can, we incorporate this one to the refined families of Sordaria and Dothidium. So you can check the publication and you can see how beautiful aquatic fungi and other terrestrial fungi also in this paper. For the classification, this is my topic. So marine fungi was estimated to be 1,500 in 1996, estimated again 10,000. This is estimated number. And in 2011, Professor Jones estimated 7,000 to 10,000 marine fungi. And from that one, it's based on the number we have in of marine fungi. So they extrapolate the data and come up with a range of 7,000 to 10,000. And here are the data that has been listed, all the marine fungi. And from 1,112, currently in what we are doing, we have 1,901 marine fungi. And you can access this one to the marinefungi.org website. We incorporate all the updates of marine fungal classification there. And when we found new species or revision of certain placement of taxa, or that taxa was raised to a higher level or rank, so we update the marine fungi. Now we have 7 phyla, 22 classes, 88 orders, and 1,901 species, but still counting. I'm still revising the number. Maybe next week or next 2 weeks, we have additional numbers. So we have 789 from the previous report. And when we compare to the estimated number of marine fungi, we only know 19% of marine fungi. And it's quite interesting. And if you wanted to be part of us that working on the classification of marine fungi and finding new species in marine environment, you can join our lab soon, maybe. I'm inviting all of you. For the marine fungi, we publish also the data on the occurrence and geographical distribution of mangrove fungi. This is very important. So the definition, because some people are asking oh, what is mangrove fungi? Maybe mangrove fungi are from terrestrial environment. Excuse me po, Mike Enriquez, sorry. So mangrove fungi refers to those marine fungi documented from various mangrove substrates. Be careful, we put marine fungi. Sometimes they're asking a lot and well, just ignore. And us collected algae, different substrates in marine environment. I discussed this in the introductory part already, so I will not discuss more. And they are, the distribution and occurrence is actually determined by different factors. For example, temperature, salinity, interaction, amplitude, etc., etc. And they are mainly composed of filamentous fungi, but we have a poorly studied data on Chytridium mycota and Mucormycota. chytrids and mucor are basal fungi. They are basal fungi because when you run phylogenetic analysis, they go down at the basal part of your tree because they are ancient. And some of them are very good, area also to study, but my friend said it's very difficult to cultivate. And we try to do more collection and he has a good morphology also. Let's see in the next few years. So these are the representative of mangrove fungi. Here, you can see from NIPA. Parang nung una, yung NIPA, ginagawa lang natin sa ating, for example, gusto mo magkaroon ng bahay kubo, NIPA. But here, we studied the morphology of aquatic fungi NIPA. So here, you can see the different fungi from different genera. This is Longicorpus. This is Striatigutula. We introduced not me, but my senior introduced this, a new family, Striatigutulaceae, from the 2 collections she had, Dr. Sheng Nanjiang. And we have also marine Sordariomycete. Marine Sordariomycetes, Dr. Devadatta, my very good senior, also studied the mangrove Sordariomycetes and mangrove Latidiomycetes. These are pictures from India. See here, these are new species. The radiella, very beautiful. They are mutiform, and as you can see, they have also sheath. This is Vaginatisspora microarmatespora, and this radiella. This is Pontoporeia mangrovi. He introduced this as a new species. Very beautiful photoplate. For the taxonomic classification of mangrove fungi, from 625, we listed 850 species of mangrove fungi under 413 genera, 161 families, and 17 classes. But most of them are composed of Ascomycota. Very few are from the basal clade. Maybe from mucor, only 1 species. Yeah, very few, less than 5 or less than 10. So these are quite interesting also because some marine mycologists doing next generation sequencing techniques, they found out that chytrids are very dominant in their data when they do the next generation or high throughput sequencing. And it's interesting, but we need to have a good morphological data also, so we can connect if they are striving there, or maybe they're just remnants of, remnants of the past, maybe. For the classes, of course, usually they're composed of Sordariomycetes and Latidiomycetes. For the order, we have Pleiosporales, the major, and for the family, we have the Halospiraceae with 94 species. For the country, in the Atlantic, we discovered, Devadatta et al. 2021 discovered 81 species only can be found in Atlantic, and the species from Pacific, 302, Indian is 206, and common species here, and here are the common species. 109 species are common to, maybe they have like geographical distribution, or maybe they can be discovered soon in the different habitats. Okay, these are the substrates. Usually, study is on submerged decaying wood, and mostly from the host Avicennia. This is Avicennia marina, followed by Rhizophora manglae and Rhizophora piculata. You can study more, read more about the different taxon. We listed them all in the current geographical distribution of mangrove fungi. You can see all the taxa there, their distribution, their substrate, and further information of mangrove fungi. I invite you to read the paper. This is a very good paper. For the freshwater fungi, the estimated number is 3,069 to 4,145. This is based on the extrapolation of data of what's currently the time. So here I'm asking myself, and we are trying to determine the known number because we already have the marine fungal numbers, right? That they are already listed. So why not list also the freshwater Ascomycota? On my paper, in freshwater fungal numbers, we listed with other collaborators 3,870 species of freshwater fungi that's already recorded based on biodiversity studies or single or multi-loci phylogenetic analysis of maybe one or multi-taxa. So mostly of them, the same with marine fungi, they're composed of Ascomycota, and you can see the new species here. A lot of new species from Sordariomycetes and followed by the Theodiomycetes. Here you can see the discovery rate of number of new species. And you can see here, it seems like we are not reaching the plateau in the identification of novel fungal taxa in aquatic environment. And it's quite interesting also, because in the Philippines, we're not exploring it yet. Maybe next few years, we can have a good data on aquatic fungi in the Philippines. Here in our paper, oh, sorry, this is different paper. This is, sorry, I did not put 22. Oh, this one, we have a good data of Chaetos spiraceae in Sordariomycetes. Yeah, found we have 823 species. And for the Deuthidiomycetes, we have 677 species of known freshwater fungi in freshwater fungi. These are some of the representative images of freshwater fungi. These are from freshwater Deuthidiomycetes. These are from freshwater Sordariomycetes. See, one thing that I observe in aquatic fungi, for example, you collected from the submerged area, right, in marine or freshwater environment, usually they burst, they shoot, because when their environment is not good, I mean, they're not used to that environment, they're already in terrestrial, so they need to release a spore so they can be widely distributed using air. And when they have another, for example, water environment, aquatic environment, the spore will attach again to the submerged wood and they can create another colony here. These are the colony. These are freshwater Eurotumycetes. These are new species from the Pseudobacterium, Tesonorea, and Veronea. These are freshwater Labulbiniales. In our paper, there are no studies on freshwater Labulbiniales, but we included that on our freshwater fungal numbers paper, and we describe the different taxa of freshwater Labulbiniales under the class Labulbiniomycetes. And it's quite interesting because there's a lot of species that more dominant than, I mean, from the genera level, from the generic level, there are a lot of species from the genera from Labulbiniomycetes. And after that one, all the information, because everything is scattered now. So what we are doing, and it's part of my work also, is to compile all the data of marine fungi and freshwater fungi from a online platform that will serve as an online repository of all the information on the classification of marine fungi and freshwater fungi. And these are the people that are currently helping us. It's not a one-man job. It's difficult, but we are trying to do our best. Sorry. So this is Professor Jones, me as a managing curator with Professor Bennett. Professor Bennett is working on the basal part of the fungus-like taxa in marine environment. And we have also in freshwater fungi, the main curator is my supervisor, Ajan Saranyapat Punmi. Ajan in Thai is like professor. Ajan and me and Sajini, my junior, we are working as the managing curator of all the freshwater fungi. And we provide description, notes, and outline of freshwater fungi. Not sure if I can show you some data, but cannot. Okay, it's okay. You can check this out. And that's all. Maraming salamat po sa pakikinig sa aking topiko ngayong umaga regarding aquatic fungi. If you have more
Ayan.
questions.
Ayan, maraming salamat po, Sir Mark, para po sa inyong pagbabahagi. So, actually, sir, meron tayong mga questions. Pero for now po, I think what would be one thing that we can actually do at the moment is to give you a couple of minutes para po uminom ng tubig bago po tayo bago po tayo pumunta sa Q&A. Meron pong mga tanong na ibinigay ang ating audience, ang ating participants. So we captured them po sa slides which we will be sharing soon. Pero sir, uminom ka muna ng tubig. So kamusta naman po ang ating mga participants ngayon? So like in the meantime, while we are waiting for Sir Mark to be ready for the Q&A, please don't hesitate to type your question po sa chat box. So both sa Zoom at saka sa ating YouTube live stream. All right.
Okay po.
And sir, I would actually grab the screen niya pala. So Mama Connie, are you there? Okay.
Yes, I am.
Mama Connie, you actually shared this paper. So let's congratulate Sir Mark din naman para dito. Ayan.
Thank you.
Ayan. Mama Connie, could you share with the audience what this is, what this
I can't see it, G.
Oh, you cannot see it?
I cannot.
I really? Like, teka. How about this time?
No.
No?
We can see it po.
Oh, why can't
Nawala sa Mama Connie. Sir, congratulations. So like I was told that this paper is actually up and coming. So would you mind sharing with our audience something siguro just lang kung ano tong publication na to. Yeah, para ma-inspire naman yung student natin that, as scientists, as you perform your research, you come up with notable milestones, 'di ba? Ito yung mga outputs kasi, no. So just to give them an idea of what's yung recent achievement po.
This one is a collaborative effort of all the mycologists that work on freshwater fungi. We come up with a list of all the freshwater fungi from 18 forgotten until now, and we provided a number. And this is very like one of my highlights in the field of aquatic mycology. We provided the numbers, the listing, the classification, and the updated taxonomic placement of the different freshwater fungi. Yeah, not much. Thank you.
Grabe, not much, no? Not much pa yun, sir. So bale makikita sa mga students, no? I think this is siguro, sir, I would just like to make a commentary about this. So kung mapapansin nyo, yung publication, it has how many co-authors, sir? Siguro more or less mga 10 to 15 co-authors.
Yes.
No? So kaya gusto ko rin tong i-share sa mga kabataan, and this was shared by Dr. Connie Gibas, is the fact that alam nyo, when it comes to research and development, when it comes to research, mabilis yung evolution ng or yung advancement ng iba't ibang areas, no? Like for example, sa aquatic mycology, the specialization ni Sir Mark, in order for you to achieve your research goals, collaborating with people could be a catalyst. So sige yan, kaya ako, sir, binabanggit yan na meron kasi sigurong medyo luma na mindset na some researchers would prefer to work on their own. Wala namang masama doon, pero if you are working in an area that is highly populated at ang bilis ng development at possible na maunahan ka ng mga tao, collaboration is key. So yun yung skill na yun, you have to embrace that in as early as possible. Yan. Kasi sir, yung atin pong audience members ay mostly high school students. So keep in mind, you don't have to be an expert in everything. And kung mag-isa ka lang mag-work, hindi mo rin pwedeng makover lahat ng bagay, lahat ng topics. So by working with experts in different fields, first, you can accelerate the progress of your work. And then secondly, you can also learn from the expertise of those people. Yan. So, ayan, maganda yan. And then, I think Dr. Gibas na offline yata si Dr. Gibas. Pero yung second naman na congratulatory slide natin is for her because very recently she's part of a team that published this article in the journal called Emerging Infectious Diseases. So just to give you an idea, so kung si Sir Mark is an aquatic mycologist, si Dr. Gibas is a clinical mycologist naman. So, 'di ba, makikita niya may representation sa international stage of mycology ang mga Pilipino. So, just to further build on yung contribution ng mga Pilipino sa area of mycology. And who knows, some of the students in the audience could be, like the future Sir Mark and the future Dr. Connie Gibas. All right. So sir, ready na po ba kayo sa questions? Sir, I think you're muted.
Ready na po. Laking background tayo.
So we were able to capture po some questions nung kanina pa sa slide. So some of them I'll be able to show sa slides, yung iba kasi bagong pasok pa lang, basahin ko lang po sa Zoom. All right, so first question. Is it possible that they I think the student who asked this question is referring to fungi. Is it possible that fungi can live in aquatic animals? So for example, fish or crustaceans as substrates?
Yeah. Usually, freshwater fungi under phylum Trigonomycota, they are basal fungi. Usually, they live as a parasite of animals or crustaceans, and most of them they are already like discovered that they can cause diseases of aquatic animals. They don't have any symbiotic relationships, but most of them are parasites of freshwater animals or even marine animals.
All right. So okay. So yan, kaparehasong talk natin last week about bacteria. So ito naman, aquatic fungi can also be parasitic in nature. Yeah. All right. Next question, sir. So ito po question naman, how long does it usually take to discover or classify a new fungal species? Kasi you show them yung different steps yung the process involved in the discovery and classification of aquatic fungi. How long does it usually take po? From the time that you collect the sample?
From you collect the sample, for the center, I mean for my own experience, maybe around, if you have molecular data already, you can introduce if it's new or not. For me, we have currently we have resources for molecular data. We can obtain molecular data of a certain species. We run phylogenetic tree. It takes maybe around, depends on the taxa, but maybe the shortest that I have is last week. Last week, I found maybe around 2 new species from the collection that I have last, what is that, 2 to 3 months? Yeah. It depends. It depends on the taxa. And it depends on what is that? The resources you have. Usually the resources. For us, we don't have any much problem. But when you have the morphological data already, for the morphological data, you can obtain around maybe 1 to 3 months, because some of the taxa, they don't have any sporulating structure in the substrate. And after that one, maybe around 1 month, you have good mycelia for morphological analysis and also for the molecular analysis. So 2 months, 3 months already, right? And another 1 month for sequencing of the necessary gene region for your phylogenetic analysis. It takes around 6 months to 1 year. Maybe that's just assumption. If you have resources.
Okay, so siguro kasi dalawa yung mycologists natin sa panel ngayon. So we have Dr. Connie Gibas. So Dr. Gibas, is it also safe to assume that the length ng process po ng classification ng fungal species ay parehas lang? Whether it be terrestrial or aquatic?
Well, as Mark said, can you hear me? Because I
Yeah.
got lost.
Yes, ma'am.
My connection got lost. So
Yeah,
I'm
we
on my
can hear
cell phone.
clearly po.
Okay. It depends on the taxa, meaning yung isolates mo, yung species mo. Kasi merong species na mabilis siyang mag-form ng structures. Merong species na matagal, 6 months, 1 year, minsan 1 week, meron na. So you cannot describe a species without structures. So depende talaga. In my experience, usually mga within 2-3 months, meron na akong morphology. Tapos, magra-write up. Yung write up, it will take almost the same amount of time. So, mga 3 to 4 months.
Apo. Okay, so eto, maganda kasi parang panel to na may alam nyo guys, ang swerte natin kasi we have aquatic mycologists and we have a terrestrial mycologist. Like eto yung dichotomy na yan is just because of their current affiliation, no? Kasi si Sir Mark nag-aaral siya ngayon. Yun yung topic niya. And then Dr. Connie Gibas, is a clinical mycologist sa San Antonio, Texas. So, since Dr. Gibas, we have talked about this, diba? And one of our main motivations before was yung frustration that you experienced na walang masyadong effort to discover fungal species in the Philippines, no? Well, in fact, diba napaka-diverse po ng ating ecosystem sa Pilipinas. So we talked about the length of time that usually takes para makadiscover, for example, ang mga students ng bagong species, kung ano yung mga gamit na kailangan. Siguro po ako napaisip lang ako. We have high school students here who might take an interest in discovering fungal species po for example sa kanilang locality. Doable po ba yun? For example, they only have 1 year. 1 year sa 4th year ng junior high. Hindi ko na alam kung ano yung grade nun. Sorry guys. Lumang curriculum ako. Grade 10 pala, grade 10. So, for example, sa grade 10 po, is it doable to at least cover a huge chunk of the work kung magsa-science investigatory project po sila?
Who first?
Sige
Are
ma'am,
you
ikaw po
asking
mauna,
me?
ma'am.
Yeah po. Kayo pong dalawa pa. Di ba?
Okay.
Ayan. So, 10 months of work. What can they do?
So, depende sa niche. So, ang ibig kong sabihin ng niche ay depende kung saan mo gustong hanapin yung fungi.
Apa.
So, katulad ni Sir Mark, ang niche na aquatic, freshwater fungi. So, medyo mahirap yan kasi maraming kailangan yan. Pero sa akin, kung ang niche mo ay leaves, plant leaves. So, doon ka lang maghahanap. Plant leaves sa lowlands, sa highlands, sa kung saan. Depende kasi makikita mo kung anong klaseng distribution. So, depende kung madali lang silang mag-isolate from those substrates. Mabilis lang. Kaya mo yung 10 months. With mentorship, with guidance. Kasi syempre mahirap ang fungi. Ang mycology is very complicated. Yun palang morphology, yung fungal biology, hindi, as sinabi ni Sir Mark, may sexual, may non-sexual, may asexual, may anamorph, may teleomorph, may silomycy. Ano ba yung mga yon? So, medyo kailangan talagang aralin mo muna yung biology bago ka pumunta sa labas. Kasi otherwise, you'll get lost.
Siguro po what would be a good follow-up po dito is basics of mycology, no? Para kumbaga to prime yung yung awareness ng mga students dun sa field para po, 'di ba? It's very important kasi you have to know the fundamentals before ka lumabas. Kasi baka
Oo.
di ba? Di ba, ano ba yung levels of classification? So siguro po Sir Mark, po, if yung question on the context po kasi isa rin sa gusto natin gawin dito is to inspire the students na mag-perform ng research, no? Ano po ba yung mga pwede nilang gawin at the high school level?
Maybe what they can do now is to have a survey of, for example, they wanted to try to observe first the terrestrial fungi. Terrestrial fungi, they're very common. So maybe for me, when I go back in the university, I collected some small specimen only in the forest, for example, and transfer and observe it on the microscope, and you can see the fruiting body. And one example that my mentor long time ago did was I practice on terrestrial fungi, because terrestrial fungi, they have a lot of fruiting body in the twigs. But for aquatic fungi, very small and slow growing. So you cannot practice properly to do cross section manually, observe the characters. It's more about familiarity of the technique. So maybe it's a little bit difficult now because we don't have much incorporation in our curriculum. What is mycology? Even microbiology. Microbiology, very elementary. Elementary microbiology. So maybe we need more like upgrade. And what we need more are mycologists that is properly trained to do
Yay.
morphological data and to have a survey of biodiversity in the Philippines.
So I like the point that you raised, sir, no, na hindi naman nila kailangan ng sobrang complex na methodologies and equipment to get started. So siguro, I like the point that you mentioned, sir, na kung meron silang light microscope and they can perform a survey, no, of yung morphology. I'm sorry, I'm a chemist, so I might not do yung jargon sa mycology or microbiology justice. Pero yun nga, just to encourage the students, no, and the teachers who are in this call, you don't have to like do DNA sequencing like in the beginning. So siguro po, and siguro po tatanungin ko lang po kasi I assume kayo pong parehas you collaborate with Philippine-based institutions. Ano po. So hypothetical scenario lang po, just to drive this discussion further. If I were a student and I did a survey, siguro po, Dr. Gibas, I would take yung hypothetical scenario mo na sa leaf, sa dahon ng halaman. Pag meron po akong isolate doon and I have pictures of the morphology, can we reach out to people like you to help to also help facilitate yung further classification? Meron po bang ganong mga mechanism available ngayon para sa mga students? And how do they initiate that po?
Yeah, marami yan. So halimbawa, first of all, kailangan mag-isolate mo na sila, or they can see the structures dun sa substrate. So, ang sinasabi nating substrate, kung saan sila tumutubo. So, in Sir Mark's yung project, tumutubo sila sa mga barks ng trees na submerged. Sa akin naman, lahat ng substrate, kasi terrestrial ako eh. Ngayon, mas specialized ako ngayon sa clinical, so mga sakit-sakit. Pero dati, hindi. Environmental din ako. Kaya kung makikita mo sa substrate, pwede mo na rin yung kumbaga, tingnan yung microscopic morphology noon. So, sisirain mo siya, squish mo siya, ilagay mo sa slide para makita mo sa microscope. And then if you can take images of that, ngayon kayang-kaya na ng cellphone, ilagay doon sa ocular ng microscope. Kayang-kaya mo yon. Kahit kung kaunting morphology na pwede nating makita, we can start from there.
Alright. Alam niyo po, na-trigger na ang mga students, binahan na tayo ng question. So, as you were answering yung questions po, I nagka-capture ako ng slides para everyone can see the questions, no, as we tackle them, para pag napanood to sa recorded version, and weirdly pumasok sila at a particular point, they can relate to, alam nila yung context. So, allow me to share. Ito, ang dami na po talaga. Teka lang po. Ayan. I will start sharing my screen again po. Okay, next question natin. Grabe, Sir Mark, mapapalaban po kayo. Ayan.
It's okay.
So let me know lang po. Can you see my screen already? Alright, so the next question po, ito napaka-basic naman ito. So siguro sabi, ang question niya po is, what culture media are used to grow fungi? Siguro po question ito is pag even you're isolating, siguro po.
Yeah. For the isolation for aquatic fungi, well, we are using water agar. Very simple water agar. You only need the water agar, put some water, and you already have water agar. These are very important in the germination of aquatic fungi because some of them will not germinate PDA or potato dextrose agar, copper rod, or malt extract agar. But if they germinated on water agar, you can transfer this to PDA and MEA because they're already producing some germ tube and they can grow properly. Yeah, but some media we use directly put PDA, potato dextrose agar, and malt extract agar. Potato dextrose agar, it's not complicated. You can just buy potato dextrose agar and create it in your own lab, maybe, if you don't have enough budget for the artificial media. It's easy, not difficult.
All right. So students, no? So there's no such thing as a super basic question. So this is your chance to ask questions like this. You know, if there are some fairly complex topics that you came across with earlier during the lecture, no, feel free to ask and clarify.
Yeah.
Ayan. Okay, sir, ang dami nito. So teka pa, pasadahan na natin to. I clicked on the wrong Ayan, ito naman po, question. So, good morning, Sir Mark. May we know what laboratory level is ideal for the culture of these species of fungi? So, freshwater and marine. And then, ang dami niya palang tanong. Are there instances too that these fungal species could be infectious to other species of organisms? So, para medyo natanong din po kanina. Pero she qualified it, marine, freshwater, and or terrestrial.
So when you say laboratory level, parang ano yun, parang what materials you need in your laboratory to provide a good description? What level, like level 1, 2, 3? But for the laboratory level, maybe for us, because we only do isolation, single spore isolation outside the laminar flow. You need only laminar flow. For the purification of your fungi, that's all. But another question, sir, species, are there instances so that this fungal species can be infectious? We are isolating saprobic microfungi. So usually they don't, for marine, usually the genera that I am dealing with are not pathogenic fungi. Maybe some, like for example, Aspergillus or Penicillium, but mostly the substrates that I collected are submerged decaying wood. So they will not give you any infection. And maybe if you submerge this to find a sporulating structure, for example, you're dealing with Ingoldian fungi and you wanted to have a good sporulation, maybe they won't infect.
Yeah. So siguro, Dr. Gibas, yes po. Dr. Gibas, this might be connected to the question last week, di ba po? Yung question about bacteria, that what would make a good bacteria behave badly? And I think Dr. Len Parungao, Balolong, she mentioned that, you know, some terrestrial microbes end up like in sa sewage, mapupunta sa dagat, and then that causes changes. Pero yun po, Dr. Gibas, do you have anything to add?
I have a comment on that one. Kasi as I am a clinical mycologist, so as I've said, I'm dealing with fungal diseases. And we are now seeing a lot of infections caused by not true pathogenic fungi, but opportunistic. So what I mean is that kung ikaw ay mahina at may napapunta sa iyo na mold or fungi, magkakasakit ka kasi mahina ang resistensya mo. So, kahit anong fungi, so katulad nun, sinabi ni Sir Mark na meron silang isolates na Cladophylophora aquatica. Sa amin, ang Cladophylophora, meron siyang mga relatives na gustong-gusto ang brain. So, I would think na kung meron kang ganon na species na related sa mga true pathogenic na fungi, I would be very, very careful. So even then, lahat kasi ng fungi na nakikita natin, ubiquitous. Ibig sabihin, makikita mo sila sa lahat, nandyan lang sila sa daan, nandyan sa mga paligid mo. Pero pagka ikaw ay humina, magkakasakit ka kung dumapo sila sa inyo. So, kailangan pa rin ng konting precaution. Okay?
So
yes, sir.
It depends on the genera, ma'am, no?
Yeah. Depende, hindi rin. Kasi katulad noon, yung ordinary na mushroom. Kinakain natin yan. Yung nakita mo, yung mga urea fungi, urea mushrooms, yung pinakita mo na maliliit na mushroom, yung pinakita mo na mushroom na nandun sa may bark ng tree na parang bracket fungi, yung Ganoderma. So, yan, Ganoderma, minsan sabi mo naging pathogenic. Pero ginagamit natin yan ngayon ng traditional medicine, di ba? Anti-cancer.
Yes.
So, paano ngayon yun? Should we be cautious about using those? Kasi nakikita natin yung Ganoderma around. Pero very, very popular yan sa Korea. Yung reishi tea. Yung Ganoderma. They're very, very good. I mean, they have very good characteristics na magiging medicinal sila. Pero yun nga, meron siyang isang aspect na naging pathogenic siya. So what makes that pathogenic? I wonder though.
For the Ganoderma, ma'am, I did not mention that
you did not?
Pathogenic. Yeah.
Oh, maybe I just I saw that somewhere. Anyway, yun.
For some of the dung mushroom, maybe some of them maybe from Lentinus. Maybe they have like but different activity. It's more of like hallucinogenic.
Yeah. Pero ang nakikita namin dito sa hospital, yung mga isolates galing sa mga patients. So they're like common na mushroom, like Tramitis, Schizophyllum, ano pa ba? Basta yung mga common na mushrooms na nakikita mo. Kaya yung kayo mga kabataan, pag kayo'y pumunta sa mga forest, be careful din. Yung hindi basta-basta na lang nagtatouch nung mga yon. If you do, be sure that you're wearing gloves para hindi dumadapo sa mga skin nyo. Yung spores ng mga mushrooms.
Ayan. So, yeah. So, diba? So, to all the students out there, this is a very good. Ako po personally, hindi naman natin ito pinanalo to, diba, Dr. Gibas? Pero we have a terrestrial mycologist and we have a marine mycologist. So, kulang na lang ay hangin, atmospheric mycologist, if that exists. Meron po ba nun?
Yung terrestrial, lumilipad yun.
Okay. So,
lumilipad yung kanyang spores.
Okay.
Kaya nalalanghap mo.
Okay, so that's very interesting then. So I'm sharing the slides again. So nakikita niyo po ba yung next question? Pero
Yes.
babasahin ko pa rin. I just wanna make sure that nakikita po. So ito po, medyo loaded din yung question. Grabe po, ang daming nating tanong, hindi ko na-capture lahat. This might take us quite a bit. So this question says, how can you start the collection of terrestrial fungi in school? How about in a lake? Any suggestions? And then, what reference can you suggest in order for us to identify the fungi? Thanks. I became interested about fungi, and thanks for inspiring our dear students and teachers. Yan.
Maybe I will answer for the lake, ma'am, and you can answer for the terrestrial fungi. For the lake, For the lake or any aquatic habitats, what you will gonna do is just to collect all the submerged one. How to determine if the submerged wood is already there for a very long time? Of course, when you break it, it's very soft and very crumbly. I mean, it's the pieces will fall piece by piece. So you can consider that one as submerged for a very long time because the color also, no, the color and what is that one? It's already decaying. So these are good substrates for aquatic fungi. But one thing you need to do more is to familiarize yourself with the structure. This is very important, actually. It takes me 2 to 3 months to distinguish what is a true fungi and from the other aspects. But yeah, because almost when I did my mycological studies in the Philippines, we do culture dependent techniques. So whatever we have during cereal dilution, we observe that one. But for this one, for the natural habitat, it will take a little bit time and techniques and familiarity to determine the aquatic fungi. But again, we have good reference. I can share you some references on how to determine if that is a fungi and if not. We have a lot. And we compiled it in one article, The Refined Families of the Thedia and Sordaria.
All right. So what about terrestrial fungi, Dr. Gibas?
Terrestrial. What was the question again?
Sampling po at the high school level, sa
Collection.
school level. How do you collection po?
Oh, okay. So dati tayong yung mga nagko-collect ng fungi, very cavalier. So, ko-collect kami ng samples, i-grow namin sa lab. Sa labas lang, walang biosafety cabinet. Pero ngayon, hindi na pwede yan kasi marami na nga ngayon. Nag-iba na kasi ang mga organisms ngayon. Environmentally, nag-change na. So, marami na silang mechanisms na that might make them not really bad because they're bad, but because they have mechanisms that may make us sick. So, kung kayo ay gustong maggawa ng ganyang project, it has to be in a lab na merong safety precautions. So, ang inaano namin, yung biosafety cabinet, kasi si Sir Mark sabi niya laminar flow hood, pero hindi kami kampante sa laminar flow hood. Gusto namin biosafety cabinet kahit class 1 man lang. Kasi baka ma-expose ang mga estudyante tapos magkakasakit sila. So liability kasi.
Yeah, which brings a very important point. So, sa mga estudyante na magpe-perform ng scientific research, whatever area it could be, whether it be mycology, chemistry, physics, safety first. So you have to make sure that, wag tayong magpaka-desperado na mag-perform ng experiment just because gusto nyo. You also have to keep in mind that your safety is much more important than the result or your research project, no? Kasi you have to make sure that you can perform the experiments, you learn from the process, but then you have to make sure that it doesn't compromise your health and safety. Yan, which na bring up naman na parehas natin na speaker. So yan. And also sa mga teachers dito, siguro ang ating panel they brought up like a very important aspect that kapag magpe-perform ng ganitong klase ng activities, kailangan may supervision ng teachers. And siguro Dr. Gibas and Sir Mark, if ever wala silang safety cabinet sa school nila, are there higher education institutions who could also assist? For example po, meron po ba sa UP ng mga ganyang groups na who could assist ang mga estudyante sa pag-handle nung or pag-perform ng isolation? Like baka meron po bang mga ganung capabilities? Ako wala kasi ako alam.
Meron.
Apo.
Meron
Yes.
sa biotech, sa
Apo.
UP Los Baños.
Yeah.
I mean, because that's where I'm from. sa Biosci. Pero sa inyo, Mark, sa UP Visayas, meron ba?
Meron po kami, ma'am. We have new regional, regional research center. Nagki-cater po sila ng mga any microbiological analysis. Tapos
that's
meron
great.
din sa UPV NIM, sa
Oh.
NIMBB. And
Okay.
meron din ngayong Philippine Genome Center. Hopefully, I think maka-cater sila kasi meron sila mga good microbiologists doon.
Kasi usually ang mga microbiologists, hindi sila nagpapagamit ng, sa akin lang to ha, kung gumagawa ka ng fungi, kung nagtatrabaho ka ng fungi, hindi maganda kung ang iyong biosafety cabinet ay ipapagamit sa microbiologist because may infect sila kasi yung spores, doon lang yun sa loob, mako-contaminate sila. Kaya dapat fungi lab, bacteria lang. Pero that's idealistic. So wala tayong magagawa kung yun lang mag-isa lang yung biosafety cabinet mo. Kailangan lang talaga sterile technique.
Yes,
Alright.
sterile technique and protected yung self mo.
Yeah.
Ayan. So again, and how do you ensure that? Kailangan yung fundamental training ninyo, no? Ayan.
Mhm.
Kailangan alam nyo yung aseptic technique.
Yes.
Sabi nga ni-
Magagawa natin yan sa susunod na talk.
Yeah, so fundamentals, yeah, Dr. Gibas will deliver that. Siguro sometime after the series, no? After nitong biology month natin. Ayan. All right.
Yeah.
Moving on to the next questions. Ang dami po talaga. I can't keep up.
That's great.
Apo. Siguro po next question. So Sir Mark, the question says, may I ask po why are Mucoromycota, I'm sorry. Hindi po ako masyadong sanay magbasa nito. And Chytridio mycota are poorly studied. And then there's a follow-up question. And then, kamusta po ang progress or development of research sa field of mycology in our country in the Philippines?
Okay. So I'll answer the first question. Poorly studied sila. Magtatagalog na lang ako. Kasi first and foremost, Chytridium mycota are difficult to isolate in culture. That's the problem. I talked to my friend here that's working on Chytridium mycota, and we collected from freshwater habitats in Thailand. It's difficult and easily contaminated. That's the problem. Or maybe what we need is a special technique more to isolate them. And for the Mucoromycota, they're easily isolated, but most of them are the same species. So I think it's more of like isolation techniques. And the problem we have right now also, there are few people working on this group. It's easy for us to observe Ascomycota or Basidiomycota in freshwater habitat, but there are few people only working. For example, in the lab, we have around 10 to 15 people working on aquatic fungi, but only one working on chytrids. And that's the problem. So we need more people to work on chytrids. And and maybe you can join our group here and work with chytrids.
Yeah.
And the
Ayan,
second,
I was about to crack a joke. So kung sino man yung nagtanong, baka ikaw na yung hinihintay diyan. Yan, 'di ba?
come.
Oo. Hashtag idea mo, research mo. Joke lang.
Yeah.
Yan, and in-invite ka na ni Sir Mark sa group niya. Oh, 'di ba? Oh, you want
yeah,
to
Ano
apply.
ba, Mark? Ang
I can talk.
chytrids ay
Chytrids,
mga zygotes yon na merong flagella. So,
Yeah.
water din sila, aquatic din sila.
Yeah.
Tapos, as you said, mahirap silang patubuin. Pero yung mga
Yes, ma'am.
halimbawa, saprolenia, yung mga ganun, tumutubo naman.
Oh.
Pero meron namang nag-aaral dito marami. Tapos sa Mucoromycota, gumagawa ako ng Mucoromycota. So like Mucorrhizopus, yung mga iba pa na like themia, usually yung mga clinicals.
Ayan.
Yes, ma'am. Fast growing sila, ma'am?
Very fast growing. Tapos kung sila ay dumako sa tao, sa amin fungal emergency yon kasi kumakain sila ng mukha.
Oh my God.
Yeah.
All right. So, okay, siguro po, very quick comment lang po dun sa second question from the both of you. I don't know if this applies kasi medyo ang hirap siguro to kasi parehas pong nasa labas ng Pilipinas ang ating experts. Pero could you make a comment on the progress and development of research sa field of mycology in the Philippines?
I think merong mga institution na nagwo-work sa mycological research. But yeah, ang problem is konti lang. So we need more mycologists sa UST, sa UPV, sa WVSU. I know, or any other institution in Manila, few people are working on mycological research. So kailangan natin, kasi island by island, archipelago eh. So we need maybe people from major islands that would work on certain taxa or certain group. So para makakreate tayo ng more of like biodiversity studies of maybe fungi in the Philippines. But sa UPV, sa pagkakaalam ko, meron kami noon na like mycobank na inalagaan ni Professor Sadapa. Kasi maraming mga fungi na na-isolate in mangroves and even in terrestrial. So maybe it's still developing. We're hoping na next timeline natin mag-okay na ako.
Okay, what about Dr. Gibas?
I have no idea. Pero I know some mycologists, but I have no idea what the extent of mycology is in the Philippines.
Okay. So siguro I would just like to make a comment as a form of
Wait po. Meron pong book na mapapublish yung group sa UPLB nila ni Kid sa UST at sa UPLB ni Dr. I forgot the name. Parang updates on Philippine mycology. Soon. I think October mapapublish.
Ayan. So siguro po I would just like to make a comment now from the perspective of fellow scientist, do ako naman po ay chemist. Sa mga estudyante, what's cool about what the likes of Sir Mark and Dr. Gibas are doing? When you discover a new species, you're creating a legacy kasi forever na yon, di ba po?
Yeah.
Hindi unless mabago yung classification, but then that's it, like your body of work will live on, forever. Kasi that's a contribution to the field already. It's very different, like say from what we do in chemistry, kasi not unless makadiscover ka ng bagong reaction, bagong chemicals, saka lang yun ma-identify with you. But then, if you actually venture into mycology, you're aside from developing technologies out of what you learn from your research, you're also contributing to yung fundamentals or yung basic science. So yun, cool lang yun. Ako parang sabi ko, parang ang saya pala maging mycologist. All right. Okay, moving on to the next question. Siguro we can entertain a couple more para hindi naman tayo mag-overlap masyado po sa kanilang tanghalian. Ito, specific question, siguro mabilis lang to. I would like to ask po, how many days, minimum and maximum, would it take to complete a phylogenetic analysis?
If you have molecular data already, right? If you have molecular data, for me, maybe one day or maybe 6 hours, I can run a phylogenetic tree with a good result. If I mean good result na ma-identify ko if new or not. Yeah. If you have already morphological data and molecular data, it's easy to run a phylogenetic analysis.
All right. All right. Okay.
Yeah,
so-
like this morning, I did that this morning. I finished one, so I'm saying I'm gonna describe a new species.
Wow.
Ay, grabe. Oy, alam nyo, yan yun. I think that's, yun talaga yung cool about this. Once you discover something new, and that's unique, yun yung sobrang exciting about what mycologists and taxonomists also do. Diba? So, yan yung, kumbaga, tinatawag na, sabi nila, sobrang bago, original. Yan. Okay, next. Can you give an overview of how you collect samples in their natural habitat, freshwater and marine? Siguro po may ipapasok ko na rin dito, sir. May nagtanong kasi kanina, kayo daw po ba pupunta po ba kayo sa ocean to collect the samples yourself?
Yes, I went to Phuket, to Wahin, very beautiful places in Thailand, just to collect some fresh marine substrates. Overview on how you collect samples. Yeah, what you're gonna do is just for a marine environment, if you want to collect in mangrove area, you need to know the low tide area, seas time, because if not high tide, you need to swim and it's muddy and there's snake, there's a lot of animals. So just for that one, you need the low tide. For freshwater, we collected usually on the submerged part, but not so deep. For example, lake, you cannot go to the inside of the lake because it's deep already. You will determine if you have a good enough samples naman eh. So hindi naman kailangan na maraming samples also because yung collection, the same species. For example, nag-collect ka this area. So parang yung population nila the same then. So what you're gonna do, if you wanted to collect, like overview, find different habitats. And pag pristine, maganda if pristine area. For example, for me, we Google the area and we find if it's far from the people. So probably you can find new species, especially in freshwater fungi. In Thailand, from 2015 to 2020, there are around 300 plus new species discovered only for 5 years. Maybe more now because there are a lot of monographs. Yeah, that's how you collect.
All right. So siguro sir, ito another question pero hindi nasa Zoom to. The question says, is there any difference in the metabolic states or pathways siguro no between terrestrial and aquatic fungi?
Yeah. Biochemical pathways, I did not explore that one, but for me, what I know is that the chemical component, I mean the bioactive compounds secreted or created by terrestrial and aquatic fungi is different. That's why more of the drug discovery is in marine environments now and in marine fungi. Usually when I studied sponge-associated fungi, they are like the second rank of most of the fungi that has a bioactive metabolites. So maybe for the biochemical pathway, maybe they have differences also because they secrete different bioactive compounds.
Okay, so speaking of metabolic pathway, since na-excite sila po sa usapan ng fungi, may nagtanong dito, gusto nang kumain ng fungi, na aquatic fungi. So ito naman po yung question. I'm sorry, nagpapatawa lang po ako. If there are edible terrestrial fungi like mushrooms that human can consume, are there also edible marine fungi? O di ba po, sobrang interested na sila, gusto nilang tikman. And if yes, how do we know if those marine fungi can be eaten po?
There are few Basidiomycota in marine fungi or even in freshwater fungi. In freshwater fungi, around 200 species, but most of them are filamentous. I'm not sure. I did not try to eat them. maybe not edible, or you need to grow them as a mycelia. And Ma'am Connie said maybe some of them may be pathogenic if you are immunocompromised. But mushrooms in terrestrial, they are so big and already studied for a very long time. Maybe that's an area, but usually we don't eat marine fungi or
Ayan.
freshwater fungi.
Okay, next question naman, ito medyo career guidance naman. I like this question. Siguro both our mycologists can answer this. Can I ask po, what made you choose to resume your profession by pursuing a doctorate degree? Bakit po kayo nag-PhD sa mycology? That's the question.
First and foremost, nung una, actually, gusto ko talaga even when I did my master's, even yung topic, I mean, all my subjects are marine biology, but I never, I never have any subjects on marine mycology. But I said, while reading all the articles, sabi ko parang konti lang yung taong nagsastudy ng marine or ng aquatic mycology sa Pilipinas. And that time, since Dr. Sadaba is an expert in mangrove fungi, sabi ko, okay, I think this is the best area kasi of course sa mga scientists dapat maliit lang yung mga nagsastudy. So dapat yun yung kinakailangan natin na pasukan para magkaroon naman tayo ng maraming taong nag-work sa ganitong field. And that time, I'm really blessed actually, kasi that time when someone asked me to do PhD and it's on aquatic mycology, and ano pa yung ina-ask mo sa Panginoon, so go lang ng go. So I decided to pursue aquatic mycology, and lucky enough na guided by the pillars of aquatic mycology. Medyo mahirap pero fighting tayo.
So siguro I will capture that kasi sir, you said you're mentored by the pillars, and this was something that offline kanina while we were setting up, Dr. Gibas highlighted. Siguro po could you Dr. Gibas, could you make a comment po kung sino yung mga mentors ni Sir Mark when it comes to the field of microbiology? I think you're muted po. Yeah.
Yung ang mentors ni Sir Mark ay yung mga who's who in mycology sa Asia and even the world. Kasi
mga sikat sila yung mga top scientists.
As in famous talaga. Kaya madikit ka man lang ng kaunti, kahit makahipo ka man lang, ayos ka na.
Ayan. So yun, by pursuing an advanced degree si Sir Mark, and also I also have to say Dr. Gibas was also trained by a very famous microbiologist who's based in Canada, and they met in Japan. You met in Japan, di ba po, Dr. Gibas? So yun, by pursuing yung graduate degree nyo, you'd get the opportunity to experience first-hand training from like the most reputable and the most famous scientists out there in your chosen profession. So yun, 'di ba? I would for example, I would just like to share yung PhD advisor ko, yung PhD advisor niya ay Nobel Prize winner. So ganun siya, yung para kang second or third generation nung academic pedigree na yon. So you just have to keep that in mind, na hindi kayo masyadong malayo doon sa mga famous scientists na nababasa nyo sa books. So nagme-mentor yung mga yan, and yun, si Sir Mark and si Dr. Gibas are examples of that.
Yeah. Kaya pag kayo ay kung mag-iisip kayo ng pursue ng grad studies, tingnan nyo yung, halimbawa, microbiology. Tingnan nyo kung sino yung mga famous talaga na names doon. Sumulat kayo. Walang mawawala sa inyo. Kasi kung hindi man sumagot sa iyo yun, hindi ka mahihiya. Hindi mo naman siya kilala. Hindi ka rin naman niya kilala. Di ba ga? So, ganun ang ginawa ko. Nakausap ko siya doon sa isang meeting. Tapos, parang ang galing nito Tapos, who's who talaga, as in royalty sa mycology, katulad ni Dr. Hyde tsaka ni Dr. Jones. So sabi ko, nung umalis na siya, hindi ko sinabi nung nagkita kami, Pero sumulat ako sa kanya. Sabi ko, we met at this meeting, tapos I would like to be supervised under you. I would like to be your mentor, ganyan. I'm very interested in mycology, kahit hindi pa talaga noong time na yon. Anyway, sinabi niya, okay. So, ganon. Serendipitous yung nagpagkakita namin. Pero, kung talagang gustuhin mo, go for it. Kahit marami kang taong sulatan mo. Baka may isang sasagot.
Yeah, so if you don't ask, the answer is always a no. So sa mga students, lagi namin tinuturo sa Philsa Hub. Ako talaga ito lagi kong tinuturo sa mga kahit sinong estudyanteng namimit ko. Kapal ng mukha is key. So kailangan yeah. Oo, yung mga mahiyain, sila yung medyo walang masyadong napapala. Kasi oftentimes opportunities hindi lang sila magfo-fall sa inyong lap. You have to pursue them sometimes. You have to put yourself out there. Yan.
Talaga, kahit sabihan kang adelantada ka. Sabi ko, ano ngayon?
Yeah. Nakuha mo yung na-reach mo yung goal mo. All right, that's thank you so much for those answers. Ito we'll entertain 2 more questions and then after that, we'll end the session kasi na-invade na natin ng tanghalian. Pero we really appreciate yung interest ng students kasi ang dami po talagang tanong. Hindi na natin ma-capture lahat. Ito po, itong nagtanong nito, 2 yung questions na. Yung una po is yung fungi sa air, which na-mention na po kanina, 'di ba, ni Dr. Gibas, that yung terrestrial fungi, sila yung lumilipad yung spores pa yung tawag din, Mama. I'm so sorry. Yeah. I forgot my microbiome one. It's been ages. Opo.
Conidia.
Opo, pero yung second question dito po yung interesting. Kasi ito, siguro ito yung laging iniisip din ng mga high school students. Paano po kayo nagka-come up ng name or scientific name for the newly discovered fungi? May pattern po ba sa pag-create ng name? Salamat po. And I was asking, kaya ako to pinick up kasi before the session, I was talking to Dr. Gibas. She mentioned to me that just today, she discovered a new species. And sabi ko, can you name it after yourself? And she said no. So pwede po bang paki-explain, how do you come up with names? With scientific names?
Ako muna?
Yes, po.
Yes, ma'am.
Ikaw, Mark.
For the scientific name, for example, you have certain genus na alam mo na, no? For example, this genus. One thing you need to consider is the character of the fungi. Ano ba yung pinagkaiba niya sa ibang species ng ganitong genus? Or maybe what you can do is ano yung area na na-isolated mo. For example, in Thailand, you can say Thailandica thailandinensis. And you need to be more careful also na yung naming ng specific epithet is magbe-base siya sa gender ng genus. They have gender. So for example, if it's a male, the specific epithet should also be a male. If the gender is female, the specific epithet is female. For example, there are a lot of revisions in the name. Or maybe ma-invalidate yung name mo if it's not good or hindi siya nag-come up sa international code ng nomenclature. Yeah, we have certain criteria for that one. For the naming of the person, you cannot name yourself and you cannot be included in the paper. For example, for us, we don't include, for example, we name to Professor Hyde, Hydicula, or Hydei, or Garrettsionensis, or Garrettsionicus. So, it should be more like respectable, and we need to find the species that will not be synonymized in the next few years. So it should be more like a very solid description of your species. For example, sa mga tao nang in-honor mo. So for me, I just want to be safe. I just name to the character of the fungi or to the area that I collected or to the environment or the location. Yeah. Or the host of the species.
O, di ba ako nagtanong kanina, natawa si Dr. Gibas when I asked her, can you name it after yourself? So, Dr. Gibas.
Hindi, ganito. Sabi ko kay Sir Jeff, Sir Jeff, pagka sasabihin ko kay Mark, Mag-describe ka new species, pangalan mo sa akin, tapos ako naman mag-describe, pangalan ko sa'yo. O, di ba? Okay yun? Pero,
Yes.
hindi talaga pwede yung, halimbawa, authority ka. So, ang tinatawag na
Yeah.
authority, pag meron kang name, so, halimbawa, ang name ko na ngayong bagong fungi ko ngayon, ang tawag sa kanya ay Kirstein Nuthelia. Mahirap na pangalan. Phaeohyphomycosum. Kasi, ang kanyang, so tatlong isolates yun na galing sa, ang itsura nitong fungal disease na to, kadiri talaga. Like, dito yun sa paa. Tapos para kang merong, ano bang tawag ng ulcer sa paa? Sa Tagalog. Yun yung lumalaki na, yung talagang sugat na nakaluwa na yung skin.
I have no idea.
Sa amin kasi hubag. Ano, sa Bisaya, hubag, di ba? Anong tawag sa inyo? Basta yun yung, pero yun yung may sugat ka sa paa na hindi gumagaling-galing. Tapos meron pang mga grains. So, ang tawag doon, chromoblastomycosis. Or phaeohyphomycosis. Sinasabi siyang phaeohyphomycosis kasi dark fungus siya. So, yung pheo, ibig sabihin dark, phaeoid. So, para kasi tatlo, para alam, kung halimbawa, titingnan mo yung pangalan ng fungus, makikita mo kaagad kung halimbawa, ang pangalan ko, phaeohyphomycosum, may idea ka kaagad, baka tong fungi na to nagkukos ng phaeohyphomycosis. So, very, very helpful, very suggestive dun sa disease na kinukos niya. Samantala, kung ipapangalan mo sa pangalan, wala kang idea. Ipapangalan mo sa, halimbawa, meron akong bagong Aspergillus americanus. Galing siya sa States, pero wala kang idea kung anong klaseng disease. Usually naman kasi ang Aspergillus, invasive yon dito sa lungs. Kaya, pero sa pangalan pa lang, alam mo na galing sa States, galing sa Amerika. So, yung mga ganyang klase.
Alright. Okay. Siguro po, last question na natin. And perhaps both of you can answer, no? From like on the context of yung source nung fungi. Can you see my screen? Yan. So siguro po, ang sabi dito, does research of fungi have economic potential? So siguro po, on the context of marine fungi and then terrestrial fungi, ano po ba yung mga economic potentials or benefits of studying fungi? So let's start with Sir Mark.
Okay. So for the ecological aspects and economic aspects, of course, when it comes to marine fungi, they are key players in the decomposition process in the marine habitats or even in any aquatic habitats. So yeah, determining the biodiversity will allow us to understand more of their ecology. And for example, if you wanted to, parehas kay Ma'am Connie, some freshwater fungi may have like the same species but can cause medical health related problems in the terrestrial counterpart. So it is very important. And marine fungi now, most of chemical marine mycologists that focus on the chemistry of marine fungi, they isolated novel bioactive compounds that can have biotechnologically what is that? Parang makakagaling. For example, certain diseases, for example, they do some any like cell blah 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 or antimicrobial analysis, and they have high potential of producing that compound. And even I think in the Philippines they're doing that now also in marine actinobacteria, and they can produce a lot of bioactive compounds. For marine fungi, you can search in the natural products. I mean, there are a lot of potential.
All right. So how about Dr. Givas naman, ano po economic benefits of fungal research?
Marami. So, as you know, fungi, pwede siyang friend, pwede din siyang foe. So, friend and foe at the same time. So, friend kasi pwede mo siyang gamitin sa food. Yung fermentation, magkakaroon ng products ng metabolites, yung mga maggagawa ng bread, maggagawa ng beer, maggagawa ng wine, 'di ba? Economically okay 'yon. Tapos pwede rin siyang from fermentation, pwede rin siyang mag-produce ng mga yung metabolites na para sa medicine. Yung aming isang isolate nga ginamit siya para yung kanyang secondary metabolite, nagpo-produce siya ng transplant rejection. So ginagamit siya para ayos yung transplantation ng dito kasi maraming transplant na lungs, kidney, lahat ng organs. So also yung kung alam mo yung fungi, kung alam mo yung biology, kung alam mo yung mechanism of pathogenesis niya, magiging healthy yung mga tao kasi alam natin kung paano siya i-manage, alam natin kung paano siya i-treat. So, economically, hindi makakasit yung mga tao, makakapagtrabaho. O, di ba ganon?
Yan. All right.
Even in plant pathology, ma'am, no? In plant pathology, management, conservation, a lot.
And veterinary.
Yeah.
Ayan. So siguro po ito to cap this session. Siguro Sir Mark, may we ask for you to siguro po send send a message of encouragement to all students and teachers participating in this session. Para po siguro ma-encourage sila to venture into the field of mycology rin, and specifically perhaps aquatic mycology.
Yeah. As you can see, yung sa slides ko kanina, that there are a lot of species na need pa natin na discover, especially sa freshwater and marine environments. Compared to terrestrial, of course, maraming tao yung gumagawa dyan. But we need more people that will help us to, determine, observe all the fungi. And sa Pilipinas or sa any other countries, Asian countries, most of the studies are concentrated sa China, sa Thailand, because they have proper laboratory, meron silang proper guidance from different, experts. And I think starting point yung DOST ngayon. Kasi binibigyan nila ng scholarship yung mga estudyante na gustong mag-aral sa labas. So you don't need to ask for the institution outside or outside the country. You need only our government institution to help you study abroad. And for us here in the Center of Excellence in Fungal Research, hindi super picky ka. Dapat matalino ka talaga. But you need to work hard. That's one character that Professor Hyde and Professor Jones wanted. If you have samples, if you have collection, collect, observe, get a good data. That's all. That's all we need. And I think katulad din ninyo ako noon. I'm just hanggang ngayon ordinary naman ako. But I think what we need is to encourage ourselves na to go out our comfort zone and ask what Dr. Connie said, ask for people na experts sa ganitong field, message them, email them, or even if you have friends. For example, my friend just have a training in Thailand that my friend Alan asked me, there's an opening of PhD, do you want? Okay, go. And I applied for the UST. So, yung point ko is hindi imposible. Walang imposible. So, everything is possible as long as sa self mo, alam mo kung ano yung gusto mo. Hindi kailangan na ang gusto ng iba. Alam gusto mo. And you need to strive and work hard for that one. And hindi siya madali. Especially when you're working on the top tier scientists. It's difficult. Even naming some species, you need to be more careful or else may mga masasabi. Yeah. Ano yan? And you need to be more like industrious also. Gusto nila yan sa mga Pilipino. We are very industrious and we can speak Hindi naman masyadong very, very good, but we are very fluent naman sa English. And that's our maybe parang edge na natin. So, sa mga high school students, kung gusto niyong magtrabaho or what do you call that one? Mag-specialize sa isang field, walang imposible. And as long as meron kang dedication sa iyong gusto.
All right. And
Thank
so
you.
thank you so much. Maraming salamat po, Sir Mark. And so with that, Filipino Science Hub would like to present this certificate of appreciation to ito medyo na-advance in Dr. Sir na, pero we will edit this, like Sir Mark Calabon from Mae Fah Luang University, MFU, Center of Excellence in Fungal Research in Chiang Rai, Thailand, for delivering the webinar Aquatic Mycology, organized by Filipino Science Hub and held via Zoom and YouTube Live on the 9th day of April 2022. Signed by yours truly and Mr. JP Onya, Head of Research and Vice President of Filipino Science Hub. So, Sir Mark, maraming salamat po sa pagpapaunlak sa aming paanyaya. I'm kitang-kita naman po sa response ng ating mga students at ng ating live participants yung interest. Ang dami po talagang tanong. We weren't even able to cover half of the questions. So, ganun po yung interest nila. So, to all the live participants out there, yun pong Google form that you have to fill out to request your certificates. They've already been posted in the Zoom chat box. So pakitingnan po also on our YouTube live chat box. And so, by the way po, biology, so April is biology month in Filipino Science Hub. So wala po tayong session next week because Holy Week yon. So we're skipping the 16th. We'll resume on the 23rd po, and it's a talk about microbiology with applications naman din sa pharma. So, see you po sa April 23rd, and then we'll cap the session. Sabi nga ni Dr. Gibas, organismal level. So, we'll talk about the Philippine carabao, its evolution, no? From a dreaded monster to say that, you know, to put it, and then evolving into an animal that could bring riches, you know, to Filipinos. Ayan. So, muli, maraming salamat po sa inyong pagsama sa amin sa isa na namang Sabado ng umaga. So, we'll see you all next time. So, by the way po, I wrote a message earlier to tell the students na merong questions. May quiz dun sa inyong evaluation form. So, dapat nakinig kayo. Anyways, alright. Maraming salamat po. We'll see you again in 2 weeks, and may you all have a great week ahead and enjoy the rest of your weekend. So, Sir Mark, by the way po, we will have a separate debriefing session po in another Zoom session. So I forwarded the details to Dr. Gibas. So muli, maraming salamat po sa lahat ng participants, to all the teachers and students out there, and sa lahat ng practicing STEM professionals so dialed in. Thank you so much for all of for your active participation. So take care everyone, and we'll see you again soon.
Until next time.
Bye.
Kapungka.
No, di ba? Kapungka. Thank you. Bye. See you po sa kabilang session.